Hello and welcome to Galaxy. Today we'll be discussing different extreme environments that certain types of bacteria have learned to survive in. The extreme environments we'll be looking at are high salinity, deep ocean, Arctic, hot springs, and space. High salinity environments are characterized as having a higher concentration of salt than water. Some examples are the Great Salt Lake and the Dead Sea. The microbes that can survive in these environments are called extreme halophiles and require 20 to 35% concentration of salt to grow. A common halophilic bacteria is cyanobacteria or blue-green algae. Cyanobacteria are known to form a dense mat-like covering over salt lakes and hypersaline lagoons. Due to the high salt concentration, the osmotic pressure is also high. This causes water to want to leave the cells. Halophilic microbes have adapted to this pressure by either synthesizing organic or compatible solutes or pumping inorganic ions into the cell. Cyanobacteria use compatible solutes to counteract the low water activity, for example, glycine betaine.
It's often a common assumption that life needs sunlight to survive and thrive, but in the deep oceans of the world, whole ecosystems manage to function without access to the sun. Clustered around geothermal vents thousands of meters below the surface of the ocean can be found a sophisticated ecosystem of organisms, and they are able to live thanks to the geothermal energy and sulfur provided by those deep-sea vents. Bacteria and archaea are able to form the basis for such an ecosystem by utilizing a process called chemosynthesis. They use inorganic compounds like sulfur as an energy source to make glucose from water and carbon dioxide dissolved in the ocean, all in the absence of sunlight. This is the method of survival for the Beggiatoa genus of bacteria, which forms around sources of sulfur like deep-sea vents in large mats.
Hot springs are one of the richest places in the world in terms of microbiology. Whilst being far too hot for humans and animals, it has a high biodiversity of thermophilic archaea and bacteria that have adapted to living under multiple combinations of extreme conditions, including hot and acidic, hot and sulfidic, hot and alkaline, or hot and filled with toxic heavy metals. This is due to the optimal growing conditions for thermophiles, a water environment and extremely high temperatures between 30 and 100 degrees with a variable pH range. The temperatures of hot springs can be categorized into 4 microbial zones. These can be seen by the pigmentation of the thermophiles exposed to sunlight. 1. High temperature zone, which hosts heterotrophic and lithotrophic bacteria that are non-photosynthetic. 2. Synechococcus chloroflexus zone. That makes up the yellow, orange, and greenish bacterial biofilm on the water surface. 3. The formidium zone, which is dominated by a filamentous cyanobacteria that forms extensive bacterial mats. And last, the calithric zone, where microbes at the top of the mat use sunlight to produce chemical byproducts, and microbes below the upper layer use these chemicals for energy and then recycle the nutrients back to the top.
Arctic environments houses groups of bacteria, archaea, and single-celled eukaryota. Within the Arctic regions, microorganisms are able to survive through many ways, whether it is harboring in the brine channels of sea ice or attached to the bottom of first-year ice and surface melt ponds. In Arctic, Antarctic, and high-altitude locations where plants are unable to survive, cyanobacteria is a primary producer of nutrients. An example is Pharodomyces priestley, a cyanobacteria of the ancient group of photosynthetic microorganisms. On the Greenland ice sheet, Pharodomyces priestley forms cryosonite holes. These are dust-filled puddles found on the ice sheet's surface. P. priestley genomes indicate it survives by producing a protective coating made from sugars.
From the depths of the ocean to the freezing Arctic, in the hot springs and the salty waters of the world, we've heard how many bacteria are able to call these inhospitable places home. But let's look up, 408 kilometers above, where the International Space Station orbits the Earth. In the microgravity environment on board, scientists have learned a lot, including how well bacteria adapt to this unique stressor. Through studies with E. coli, it has been found that bacteria can thrive in microgravity when compared to controls on Earth, with an increase in the final cell count, the thickening of the cellular envelope, and enhanced biofilm formation. These studies have also shown an increased resistance to stresses such as